Hello, everyone. This is Sina Majidian. I'm a postdoc in Christophe Decimo Lab in University of Lausanne. Our lab hosts OMA browser, a well-established resource for orthology inference. Today, I'm going to describe a major update to our software, OMA. Let's begin with the definition of orthology. Consider we have two species here, human and chimp. Each of them has two proteins. And consider there was an ancestral gene A, and during a duplication event, we had two A1 and A2 genes, and during a speciation, we will have these four genes here. And the definition of orthologs is that when two proteins in two species evolved from one gene in the same common ancestor, diverged from a speciation event, we call them orthologs. For example, here, CA1 and HA1 are orthologs. Similarly for CA2 and HA2. On the other hand, these CA1 and CA2 diverge from a duplication event here, so they are paralogs. And similarly for HA1 and HA2, they are paralogs. These kind of information are informative to study gene evolution and inferring phylogenetic trees. But the challenge is that they are usually found using all against all comparison, which is not scalable. In order to solve this challenge, we come up with the idea of using a new concept called hierarchical orthologous groups that can be defined as sets of genes descended from a common ancestral gene at a specific taxonomic level. To describing this concept, I'm using this visualization. Consider we have a species tree for human, chimp, and mouse, and there were two rounds of speciation, primate speciation, and mammal speciation. And this is our gene tree. We have these five genes. And let's start with primate level. At each taxonomic level, for example, starting from primate, consider these four genes. These two genes descended from this common ancestor, so they can be grouped together as a one hog or sub-hog. And also these two together descended from this point. But this node in the gene tree is a duplication event, so we need to have two different sub-hogs. Let's go one level deeper to mammal speciation. We have all of these five genes together because they descended from this point as a common ancestral gene. So we can have this nested structure which can be used to parallelize our approach. Our tool called FastOMA starts from proteomes, sets of proteins that we map them to our database of gene families or hogs using a very fast k-mer approach. Then we have these gene families or root hogs. For example, here root hog 1, root hog 2. And the positive point of this approach is that we don't need to compare gene family 1 and gene family 2. We only need to do it inside each gene family, so we can highly parallelize our approach. Then we infer sub-hog or each different events for each taxonomic level in parallel, thanks to the hog concept. Then we will have the hog information, which implies the orthology relationship, and we can benefit from it. To make it more details, again, the same example, we have these five genes or proteins, and at the leaf level, each gene is a sub-hog itself, and we want to do a bottom-up approach. So we start from the leaf and goes towards the root of a species tree, and we want to decide to merge some of these hogs from the leaf level. Sorry. Yeah, let's go one level deeper. At primate level, I want to decide which of these four should be merged together. First, I used a multiple sequence alignment. and then I infer the gene tree, and I need to root it, and then infer the speciation or duplication event using a species overlap. And here I will have this gene tree. Then using the speciation event in this gene tree, I decide to merge these subhugs. For example, here, this field one should be merged together. And I will have the subhugs for primate level. Then again, I can go one level deeper to mammals. And here mammals, and I decide whether to merge these subhugs or not. And then finally, I will have the, because of this speciation node, I will have the subhugs at the mammal level. So I can show different levels as a nested structure, which is hug, and we can report it as a standard way called OrthoXML format with different groups that are inside each other. So to benchmark our method, we use a QFO, Quest for Orthology benchmarking dataset. And here we can see FastOMA in black, which is on the Pareto, and the results are quite comparable to the rest of method. This is the OMA, groups, which is with low recall in this species discordance test. The idea behind this test is that it compares the gene tree with the species tree, and we calculating the average RF distance as the precision. So the lower the distance, the better. And here the recall shows how many trees are sampled. Another benchmarking set is the comparing the orthology pairs inferred from the reference phylogeny. Here we use Swiss tree, and we can see that FastOMA has a good precision and comparable recall compared to others, state-of-the-art methods. So this was the accuracy. Now I want to show how fast it is. Usually, many of the orthology methods can scale to 10 or hundreds of species. Here, we were able to run FastOMA in a single day using 300 CPUs. This is 2,000 species. And the current version of OMA is a bit slow, can only handle tens of species. And OrthoFinder, the standard version, was a bit challenging, and using the parallelized version needs several CPU hours, and at some point it failed with out of memory. So our method is implemented in Python and is ready to use in our GitHub. And in summary, FastOMA is a scalable method with comparable accuracy. Thanks for your attention.
Thank you. I think we have time for one question.
Thanks. Congrats on improving this. It was not so fast before. What is the size of the k-mer that you use?
So by default, we have a method called OMAmer that was published in Bioinformatics a few years ago. The default value is 31, if I'm not mistaken.